Hey everybody, this video is about mutations. Okay, so you may have heard of mutations before. If you've seen a superhero movie, you've heard that some people get mutations that give them superpowers. Well, in reality, mutations are typically much more subtle, and they're not always good and they're not always bad. Sometimes they can be beneficial, sometimes they're not. But a mutation is just a change in the DNA, and that change in the DNA eventually affects the traits that get expressed on or inside of your body. So if you remember, people's traits are controlled by proteins. So whatever kind of proteins that you have in your body are going to determine what type of traits you have: hair color, skin color, hair type, hair thickness, eye color, all these things, okay? They're all controlled by the different kind of proteins that you have. And proteins are made up of amino acids. And the amino acids that your body puts together in that certain combination is controlled by the instructions given by the stuff called mRNA. The M stands for messenger. And the instructions that the mRNA has are just a copy of the original instructions that exist in your DNA. So we've talked about this in the past, how you have instructions for how to make proteins coded into your DNA. They get rewritten into mRNA, which is transcription. That's step one of protein synthesis. That mRNA instructs which amino acids get put together and in what order to make the specific protein, which controls your trait. Let's say hair type, curly or straight, right? Okay, so what a mutation is a change in the DNA. Once again, it's not always bad. Sometimes that can be good. But if we change the DNA, then we will change the mRNA, because it's just a copy of the DNA. And if we change the mRNA, we'll change the amino acids. The type of amino acids or the order that they go in to make the protein. And if we change the protein, we'll change the trait. So a mutation is a change in the DNA that leads to a change in the final trait that you express. So your DNA, though, what really makes one protein different from another is the sequence of nucleotides. Remember, DNA is made up of these things called nucleotides. DNA is a long chain of molecules. So we have a chain here. The whole chain is DNA, but each link in the chain, so one of these guys, that's the nucleotide. So all these are nucleotides linked together and they make up DNA. And there's actually two strands of nucleotides in DNA. As you remember, it's double-stranded, not single-stranded. Okay. But these nucleotides are actually, if you'll remember from a previous lesson, made up of three components. There is a phosphate, a sugar molecule, and a base. Now, the phosphate and the sugar are always the same, but remember, in DNA, this base will be either a G, a C, an A, or a T in DNA. And in RNA, it's GCAU, right? We learned about that previously. So it's always going to be phosphate sugar G, or phosphate sugar C, or phosphate sugar A, or phosphate sugar T, okay? But we don't want to draw phosphate sugar G, phosphate sugar C. We don't want to do all that. So what we do instead is we just write the letter. G, C, C. I'm just going to write random letters, okay? T, G, okay? So let's say that this is our DNA sequence. Now this is just one strand. This is strand number 1. DNA has a second strand, and across from it will be C, G, G, T, A, C. You remember your base pairing rules. A always goes with T, C always goes with G. That's why they're always across from each other. Okay? Now, let's say, but remember when we read the protein, or when we make the protein, we're only going to read one strand of DNA. So let's say that this is the strand of DNA that we are supposed to have, CGGTAC, okay? This is the strand that we're going to read. So let's just get rid of this guy altogether. And let's just look at the strand that we are going to be making a protein from, the sequence that we're going to be making a protein from, okay? All right, so this is DNA. If we rewrite the DNA as mRNA, then we're going to rewrite it with its complements using GCAU. So C always goes with G, G always goes with C, G always goes with C, T always goes with A always goes with T normally in the DNA, but it's RNA, so it's U, C always goes with G, okay? Then we break these up into chunks of 3 that we call codons. So this codon will make a particular amino acid. This codon will make a particular amino acid. All right, so what does this codon make? GCC, we go to our codon chart. GC, by the way, this is a different type of codon chart. You find the first base here on the left side. You find the second base on the top, the third base on the right. So you go left, top, right. So it's G, so you know it's going to be in this row. C, it's going to be in this column. And then C, alanine, okay? So we know that's going to make alanine. AUG, I can already tell you what that's going to be because that is a start codon. AUG, which is methionine. Okay. All right, so we're going to put MET for methionine. So this is our tiny little 2 amino acid protein. That's an amino acid, that's an amino acid. They link together, the whole thing is a protein. Okay? So this is what it's supposed to be. This is how it should work. Now, let's say though that we have a mutation in the DNA. Let's say that instead of there being a G here, let's say that there is an A. So that's the mutated base. We took the G out and it's been replaced by an A. So we have changed the DNA, we have caused a mutation. Now, instead of this being, well, C will still be G here, but instead of this being a C here, because this is an A, this will now be a U. Okay, so everything else stays the same, we're just changing this letter because this letter is different, so when we rewrite it, it also changes that. Change the DNA, you change the RNA. You change the RNA, you change the protein. So let's see what GUC makes. G, U, C, valine. So we now have a different amino acid, valine. And because we have a different amino acid, we now have a different protein altogether. So when we change the DNA, we change the RNA. When we change the RNA, we change the amino acids that make up the protein, thereby changing the protein itself. This is now an entirely different protein. Okay? So instead of making your hair curly, it could make it straight. Or instead of it being straight, it could be curly or something like that. Okay? There's more. So there's different kinds of mutations. So what we just saw here, when we replace the G with the U, this is what we call a substitution. So we substituted one letter for another. Okay? It's like a substitute teacher. You substitute one teacher with another. So that was a substitution, what we just did there. But there's also other types of mutations. There's what's called an insertion. And an insertion, this is when a letter gets shoved in, or a base gets shoved in to where it shouldn't be, okay? So we're cramming in, let's say this G over here gets crammed in between these guys where it shouldn't belong. Now what that does is it makes that a codon. Well, this will eventually become a codon, and then these three, and then you have this one extra. So when you change that, you're going to change the mRNA, and you're going to change which codons are what, and each codon makes an amino acid, so you're going to make different amino acids, and now you're going to make a different protein altogether, and it could mess up your traits, okay? So that's an insertion, when you put a letter in, and it shifts everything down one. Or there's a deletion, you can do the opposite, where instead of there being a letter added, there's actually a letter taken away. So let's imagine that this never happened. Okay. And a deletion is just where a letter gets completely removed. And now you're missing a letter. So now there's nothing here, and if there's nothing here, then you can't make an amino acid at all. The amino acid just disappears. And now your protein is broken. You don't even have a functioning protein. Okay, so that one tiny little change, whether it be a substitution, where we replace a letter, or an insertion, where we cram another one in, or deletion, where we take one out, all these things can have really big effects on the end result of the protein, which is what controls your traits. Now, mutations cannot be passed on. They can't be passed on to your offspring. Some people think that if you have a mutation, your babies will have a mutation. Not necessarily. It's possible, but not necessarily. Okay? So, let's say you have, well, I have to tell you this first. So there are what we call somatic cells and gametic cells. Gametic cells are sperm and egg. Everything else is a somatic cell. Okay? If it's not a sperm or an egg, it's a somatic cell. Only sperm and eggs are gametic cells. Now, if you have a mutation in the DNA, because your sperm has half of a full set of DNA, and then the egg has the other half of the DNA. It's like if you have a bunch of pairs of shoes. Well, the egg has all the left shoes, and the sperm has all the right shoes. And when they come together, they make all the pairs of shoes that you should have, okay? But let's say that either the sperm or the egg have a mutation somewhere in their half of the DNA. Well, then, when they join and make the baby will have the mutation. Because the sperm or the egg that made the baby had the mutation. But, if mommy and daddy have a mutation, but it's in their somatic cells, which is anything that's not a sperm or an egg, well then, they will not pass on the mutation to their babies. So there will be no mutation passed to offspring. All right, so that is it. So just keep in mind a quick recap what we talked about. Mutations are a change in the DNA. If you change the DNA, you change the RNA. If you change the RNA, you change the protein. Change the protein, you change the trait. 3 kinds of mutations: deletion, insertion, substitution. And they can only be passed on offspring if they happen in the gametic cells, not the somatic cells.